the smallest unit of life is just one cell big. This means that cells are the key to life. A single cell has everything required for life. Now, if we look inside cells using microscopes, we can see that they are in fact compartmentalized into lots and lots of different individual structures, which each have their own individual role, but work together to help the cell carry out its various functions. In this section, we're going to look at the structure of eukaryotic cells and how to observe them using microscopes. So all cells can be split up into two categories, eukaryotic cells and prokaryotic cells. The major difference between them is that prokaryotic cells do not have a nucleus. Now in this section we're going to be learning about the structure of eukaryotic cells, and later on in the course you'll be covering prokaryotic cells. In 1665 the biology world changed forever because the first microscope was invented. It opened a whole new and exciting microscopic world which allowed scientists to study cells and extremely small organisms. Ever since then, the microscope has developed, and today it is a very sophisticated piece of technology. We're going to be learning about two types of microscope, the light microscope and the electron microscope. So the man who invented the first microscope was called Robert Hooke, and the microscope looked like this. He looked at many different things down his microscope and he drew them in amazing detail. Here is a picture of a flea that he drew. Now one of the things he observed down his microscope was this. Now, this is a piece of cork, and he saw the little compartments and he thought they looked just like the cells that monks lived in. And so he decided to call these little compartments cells, and this is where the term originally came from. After plenty more research, it was finally realized that cells were the basic unit of life. And in 1839, Schleiden and Schwann coined the cell theory, which stated that all living organisms are made of cells. Microscopes are now much more advanced, and a huge amount has been discovered about how cells work since then. There are two types of microscope we're going to be looking at, the light microscope and the electron microscope, and we'll be looking at the various strengths and weaknesses of both these types of microscopes. Now in terms of magnification, how much bigger they can make an image, the light microscope can magnify an image by 1,500 times, and the electron microscope can magnify an image by 500,000 times. It's a very powerful microscope. However, it's not just about how big an image is, but how detailed the image is. Now that is down to the resolution or the resolving power of the microscope. Basically what this means is to what level of detail can the microscope distinguish two points that are close together as being 2 points close together, or do they blur and become one? In these images you can see they are magnified at the same level, but one is at a higher resolution than the other one. Now, the light microscope has a resolving power of 200 nanometers. So that means if there are 2 points that are 200 nanometers apart, it will be able to distinguish them as 2 points. Any closer than that together, and it would blur and become 1 point. The electron microscope, however, has a phenomenal resolving power of 0.5 nanometers. Now a nanometer, we're going to become more familiar with the different units used in cell biology, but a nanometer is a millionth of a millimeter. So a light microscope works by light entering at the bottom. It may come from a lamp that's built into the microscope, or it could be an external light source that bounces off the mirror. The light passes up through something called a condenser, which concentrates the beam of light. It then passes through the specimen, which will be mounted on a glass slide on top of this part of the microscope called the stage. That beam of light will then pass into the first lens, which is called an objective lens. Usually there are 3 lenses that you can rotate round which have different magnification, and this is what's going to magnify the image and make it bigger. That light will then pass up through another lens at the top called the eyepiece lens, which will magnify the image one more time before it's passed into your eye. To work out the total magnification, you just multiply the power of the objective lens with the power of the eyepiece lens. Now, in order to prepare a slide for viewing on a light microscope, you'll need to make the sample extremely thin so that the light can penetrate it. Otherwise, you won't see anything. And also you may need to stain the slide so that certain structures are easier to see. For example, hematoxylin and eosin stain is used to stain the nuclei pink and the cytoplasm of certain cells. Methylene blue stains nuclei blue in animal cells, and iodine is a common stain used to stain plant cells. Now, some really good things about the light microscope: it's very cheap, very accessible, very portable. You can look at live cells without very much preparation, so you can look at blood and semen samples very, very quickly and see the cells still alive in their natural states. What's bad about it? Well, it's not really a negative, but it's, in comparison with the electron microscope, is that its magnification resolution is limited. You're only going to be able to see structures of a certain size with a light microscope. Now, the electron microscope works on a similar principle to the light microscope, but instead of a beam of light, we're going to use a beam of electrons instead. Now, an electron beam has a much smaller wavelength than light, and that means we can magnify it higher and that it will have a higher resolution. The beam isn't focused by lenses in the same way because it's not optical, but it will be focused by magnets. There are two types of electron microscope: the transmission electron microscope, or TEM, and the scanning electron microscope, or SEM. So the transmission electron microscope produces images with the highest magnification. The images are two-dimensional, but they have a higher resolution than the scanning electron microscope. You tend to look at the internal structures of cells or organisms using a transmission electron microscope, and the images are produced on a fluorescent screen at the end. Now, the scanning electron microscope is different. You don't get as high a magnification or resolution, but you do generate some particularly nice 3D images because it focuses on the surface and can only show the morphology of samples. It works by measuring electrons that get bounced off the surface of the sample that you put in, and the image is generated on a monitor. And you get some particularly nice images. For example, these are all different types of pollen, or here we've got some bacteria. Now, the colour is actually fake. The colour is added by a computer at the end. This microscope can only actually generate images that are black and white. Specimen preparation for electron microscopes is quite complicated. It involves three sort of stages: fixation, dehydration, and then sectioning. Now, for fixation, the sample must be fixed with certain chemicals. This is because a beam of electrons has such high energy that it can get up to a temperature of 150 degrees C, and that will destroy most of the structures that you want to see. So they need to be fixed with certain chemicals. Now, the problem with that is it does create unwanted artifacts in the final image. You then need to dehydrate the sample, because in order for the sample to be viewed, it needs to be done in a vacuum. If you fire electrons when it's not in a vacuum, then the particles of the air will ruin them, and it won't generate an image at all. So we need the sample to be done in a vacuum, but water boils in a vacuum. So the sample must be completely dehydrated and removed of all the water before you can view it in an electron microscope. Now, in terms of sectioning or coating, it depends whether you want to look at the sample in a transmission electron microscope or scanning electron microscope. The transmission electron microscope, you need to embed the sample in resin and slice it into ultra thin sections that are about 100 nanometers. The scanning electron microscope, you actually want to coat the outer layer of the sample in a very thin layer of gold or palladium, and that allows us to generate this 3D image of with electrons being bounced off the surface. Now, the good things about the electron microscope is that they have a huge magnification and a really, really good resolution. Now, the bad things about the electron microscope are that it is complicated to use. The samples need to be dead, the samples need to be dried out, it needs to be done in a vacuum, it's very, very expensive. They are a large piece of apparatus, you need a lot of skill to prepare the samples, and you're very likely to get artifacts at the end, so it's quite hard to interpret the images. When you compare that to the light microscope, which is so quick and easy to generate an image, you can see why both types of microscopes are still used. Now, you must be able to calculate the magnification or actual size from a micrograph. Now, a micrograph is the name of a picture that has been generated through a microscope. To do this, you're going to use the I am triangle. I stands for image size, A for actual size, and M for magnification. So here's an example for you. Here's a bacterium which is being viewed at 20,000 times magnification. And we want to work out the actual length. So if we use our triangle to work out the formula, we're going to use actual length equals image size over magnification. Now you're told the magnification, so all we need to do is work out the image size, and all you have to do to get that is to get a ruler and measure the size of the image. Now in this case it measures 8.2 centimeters, so we take 8.2 centimeters and divide that by the magnification of 20,000, which gives us the actual size of this bacterium is 0.00041 centimeters. But that's not a particularly useful format to have the number in, so we're going to convert that into micrometers, and that will give us 4.10 micrometers. That is the size of this bacterium. For these questions you will need to be able to convert numbers into different units very easily. So the most common conversion you'll have to do is between millimeters and micrometers, and there are 1,000 micrometers in 1 millimeter.